Today I'm going to talk to you about something that we've really been interested in since I started a lab, and it's about brain arterial venous malformations, and actually has a collaboration with another former postdoc at Gladstone, Jason Fish, who's now at Toronto. So before I started, though, I just want to say, it really was see if I can adjust this a little bit, an awesome place to do science with some really great people. And over the years, there's just some wonderful colleagues and teams that Benoit assembled. And just as importantly as the career stuff, is really a great group of people to have fun with. And I think that was the thing, is like it was super enjoyable in and out of the lab. And I don't know if that's normal to have that much fun after hours with your team, but that was kind of it. It was like Thursday and Friday and Saturday, you knew it was like, all right, Friday, you're probably going to go dancing, whether it's in the Castro with John and Natalie, or you're going to catch a baseball game, or, we'll like lab did a lot of trips together. I think this is like my future wife with Nina when she was just a little baby up in Napa, Natalie's daughter, and it's just a blast. Whether happy hours that actually lasted way, way too long or other things. So it's a great place to be there. So I'll get to the science. So arteries and veins are separated by capillaries, and this is the normal case of where oxygen and nutrients are exchanged, and this is really the business of the endothelium. And however, arteriovenous malformations, or AVMs as they're called for short, are the result of direct connections between arterial vessels, which are high flow, and low resistance capacitance venous vessels. And these are one of the most difficult to treat vascular anomalies, and they're associated with high morbidity and mortality. Now, one of the most dangerous and complicated type of AVMs are found in the brain, and those are known as brain arteriovenous malformations, or BAVMs for shorthand. And these are usually unifocal, meaning that there's one lesion per patient, unlike some genetic syndromes like HHT or CMAVM, which patients can have multiple malformations. And the symptoms that can bring a patient into the clinic are anything that's as benign as a headache or a seizure, or something as severe as hemorrhage or stroke. And basically, what drives home the severity of this disease is the patient's risk of having a brain bleed is 105 minus a diagnosis. So basically, if a 20-year-old gets diagnosed, they have an 80% chance, or 25-year-old has an 80% chance of having a brain bleed at some point in their life. And 29% of those patients with a rupture will die, while 55% will lose independence. And up until some work that we've done with our collaborators, there's really no understanding the genetic basis of this, because the majority of BAVMs are not inherited. So what we had done, being pulled in by Jason Fish's team, and in a study led by a very talented neurosurgeon, Ivan Radovinovic, at Toronto Western Hospital, is aided in informatics by Sergei Nikolov, who now has his own lab in France. Is they had taken brain arterial venous malformation tissue that they excised from patients during surgery, and then compared it to peripheral blood controls. And after whole exome sequencing and looking for somatic variants, again, because this is not an inherited disease, and this is published in 2018 in New England Journal of Medicine, the team was able to discover that most of the patients, about 50%, had mutations in KRAS. And in a replication cohort provided by Johanna Frozen at Kuopio University Hospital in Finland, we saw that this was able to be repeated. And again, about 50% of BAVM, or brain arterial venous malformation patients, they were able to detect mutations within KRAS. So importantly, we were able to find through magnetic activated cell sorting for endothelial cells. So we purified endothelium away from all the other cell types in the brain from these lesions, and did digital droplet PCR. We're able to show that in whole tissue, compared to the endothelium or CD31 or PCAM-enriched cells, that there's a low variant allele frequency of about 0.5% up to about 5%. Most of our bulk tissue that rose from 5% to about 45%. That the endothelium was a tissue in these lesions that harbored these KRAS mutations, as we were unable to detect mutation in any other cell type like fibroblasts or smooth muscle cells. So this basically raised a question of, okay, well, what is KRAS and how could it be causing these malformations in the endothelium? So for those of you unfamiliar with KRAS, the RAS family of proteins are binary molecular switches cycling between an inactive GDP bound state and an active GTP bound state. And receptor tyrosine kinase mediated activation of the GEF basically drives it towards an activated GTP bound state, where then GTP bound RAS is able to act on its numerous downstream effectors such as RAL, GDS, PI3-AKT signaling cascade, MEK-ERK signaling, as well as others that are also not shown here. And the upshoot is it can affect processes such as membrane trafficking, cell survival, and calcium signaling, apoptosis, as well as a host of other things that are not shown here, affecting proliferation, differentiation, migration. And importantly, basically the stop for the signal is a reversion mediated by GTPase activating proteins or GAPs. This is essential so that you can basically cycle this switch on and off. Now, the consequences of the mutations that we had actually identified in our patients have been known for decades in the cancer field to lead to constitutive activity of KRAS. In fact, they're preventing GTP hydrolysis and this being shut off. So these mutations that we identified, like G12D, V, and Q61H, all prevent GTP hydrolysis, and then you would expect can lead to constitutive activation of one or all of these downstream effector molecules. So the $1 million question that we had is, okay, now we've diagnosed this in the patients, is this mutation really causative and able or sufficient to cause AVMs in the brain? So this is work led in the lab by a former postdoc, Alexander Herman, as well as a graduate student, Carlos Flores-Suarez, and a wonderful technician, Karen Bermudez-Ruiz. And what we did is we used a earlier allele that Tyler Jacks had generated, where they knocked in a stop cassette to endogenous KRAS locus. So this is not an over non-physiologic level of expression targeted to ROSA or something. It's basically if KRAS is not expressed in the normal cell type, this transgene will not be active either. So in that endogenous locus, they knocked in a stop cassette upstream of the mutant exon, this constitutive G12D mutation. And in the presence of CRE, then you would have the normal promoter driving this. So the strategy that we took was to use a somatic endothelial cell expression with postnatal onset by giving tamoxifen. And what we saw, though, is, in the interest of time, I'm going to tell you, not show you, is that this led to lethality. So basically, we pushed this system to the background and focused on instead a brain or central nervous system endothelial cell specific CRE driver, again, using a somatic or postnatal specific induction regimen. And this is what we had. So we mated this specific CRE, SLC, a 1C1 or CNS endothelial CRE, cross this KRAS gain of function model, give tamoxifen P1, and then see what happens to those KRAS mutant overexpressing in endothelium only mice. And for our first analysis, what we've done is actually take fitzy-lectin, first perfuse it in the tail to hit the veins, and then at harvest time, perfuse it through the heart, then take PBS, image the brains, and also then subject the brains to clarity clearing and light sheet microscopy. This is an example of what you'd see from the olfactory bulb to the cerebellum, a really nice labeling of the arterial territories in the brain. This is pretty much how you expect it to look in a mouse and adult, and we're looking kind of top down onto the cortex. So if we zoom in here, and even more so, you can see nice arterial labeling there and normal penetrating arterioles going down into the brain. If you see light sheet microscopy here in 3D visualization, you can basically see that there's no odd shunts or tortuosities, and you have a normal capillary network. And this is exactly what you'd expect it to look like. We're just flipping around to the underside there. You can appreciate that this overall looks fairly normal. However, in our endothelial central nervous system or brain-specific KRAS gain of function mice, if we zoom in here to the cortex, you can see here that there's this large abnormal vessel. If you zoom in here, what I hope you can appreciate is this artery is connected to this large venous dilation that's diving under there. There's also several tortuous or twisty-looking arterioles next to it. So if we see here by light sheet, a higher resolution, this time coming from the bottom, flipping over towards the top of the cortex, you can see that there's really this large tangled nidus or shunt here between, again, the dilated vein or draining vein and this artery branching off from the MCA. So this to us is really satisfying, demonstrating that not only is KRAS mutations correlated with BAVMs in humans, but in mice it's actually sufficient to drive this malformation in the brain. So then a question became mechanistically, how does KRAS drive these AVMs in the brain? And there's numerous options, as we said, that KRAS GTP can act on any one of these downstream effector pathways. So some work that Jason did in the lab, which is really nice in that initial New England Journal paper, is they queried these various downstream effectors for activation within the AVM endothelium or CD31 positive cells, as well as normal vessel positive cells from a lobectomy from a epilepsy patients. What they saw is upregulation of ERK phosphorylation in the AVM endothelium, but not in the normal vessel endothelium. And we saw no difference in phospho-AKT nor in P38. And they didn't survey the other downstream effectors, but basically this suggested that somehow or some way that active GP loaded KRAS or active KRAS was preferentially acting on the MEK-ERK pathway in endothelial cells. So essentially, this focused our attention on this part of the pathway. So then what we noticed was really interesting is we took HUVEX, transfected with dominant active G12V KRAS 4A or 4B, and in the absence of serum, then we did RNA-seq, and we compared this actually to some HUVEX that we'd grown in the presence and absence of VEGF for an earlier paper that we'd published. And through gene set enrichment analysis, we saw a really nice correlation between the genes that are positively and negatively regulated between VEGF growth factor stimulation or KRAS dominant gain of function. And this was basically leading us to suppose that potentially in a normal setting, you might have VEGF and VEGFR2 going through PLC and PKC to potentially act on the MEK-ERK kinase cascade. And this was a model built by previous papers that we published in the lab. And then we reasoned that potentially KRAS was hijacking that pathway to lead to constitutive activation of this angiogenic network, which potentially was what was driving arteriovenous malformations. So this is kind of the model where we were at up until this paper we just published in Circ Research. And if this model were true, we reasoned that MEK inhibition should be able to ameliorate or rescue those arteriovenous malformations or gene expression differences. So what we saw is if we took HUVEX, again, plus and minus dominant active KRAS in the presence of MEK inhibitor or PI3 kinase inhibitor, again, another downstream effector that we didn't see upregulated, this is as another control, and did RNA-seq, what we could see is that KRAS upregulated about 350 genes or so, 400 genes, and of those KRAS up genes, about 150 were actually MEK dependent. When we look at the gene ontology analysis, you can see that many of the processes such as VEGF signaling and angiogenesis and cell migration were MEK dependent. So basically KRAS-induced genes for these processes required MEK activity. However, when we looked at PI3 kinase sensitive genes, again, these are KRAS upregulated genes that were downregulated by PI3 kinase inhibition. It was only sufficient to give us 2 genes in one category with a low FDR. So it really wasn't effective. And it suggested to us that the majority of the KRAS-induced transcriptional changes in endothelium required MEK activity for their angiogenic response. So that was great. But again, it's RNA-seq in a cultured cell. So what we decided to do is to test this model using mutant KRAS, reasoning that if it activated the MEK-ERK kinase cascade, it would stimulate angiogenic behavior, and potentially using a MEK inhibitor like U0126 should be able to block these effects. So what we saw initially is that if you take HUVEX and transfect them, again with dominant active KRAS, normally you see nice V adherent expression here, and in the presence of KRAS, they disassemble their junctions, and they also upregulate ERK phosphorylation. However, what Jason's team saw is that they added the MEK inhibitor, you're able to restore that junction formation and downregulate ERK phosphorylation. So again, this is in a dish, but very suggestive and in agreement with the RNA-seq data. So then we move this to an in vivo system, zebrafish. We took an endothelial promoter driving a GFP dominant active KRAS fusion and inject it into a transgenic line that has erythrocyte labeled DS red. So basically every blood cell is going to be RFP positive. So it's kind of like an in vivo angiogram assay. And we looked for shunts. So we saw in our uninjected controls So at 36 hours, this is anterior to posterior, the top vessel's artery, the bottom vessel's the vein. And if you look at 36 hours post-fertilization to 60 hours, you see a really nice remodeling and a clear artery vein distinction between these two vessels. However, in our KRAS gain of function endothelial-specific zebrafish embryos, you can see here that there's a shunt here that by 60 hours, it's very clearly short-circuited as the vessels don't go all the way to the end of the tail, and they have this premature short-circuiting. However, if we add MEK inhibitor after 36 hours in fish that clearly have shunts, what we saw is that SLC327, again, a MEK inhibitor, could actually rescue or restore that nice arteriovenous pathway. So this showed in vivo that MEK inhibition could actually rescue KRAS-driven arteriovenous shunts in vivo in zebrafish. So today, in this short time, I hope to convince you that we identified a pathway whereby dominant active KRAS gain of function in the endothelium leads to activation of the microRNAs cascade in vitro and an angiogenic transcriptional signature in cultured cells and arteriovenous malformations in zebrafish and mice, and that inhibition of MEK in zebrafish will rescue those arteriovenous malformations. So with that, I need to thank the people who actually did the work in our lab. Carlos Flores Suarez took over the project for Alexander Herman, who now is a agent in the FBI, and is aided in the informatics and light sheet analysis by another talented graduate student, Manuel Cantu Gutierrez. And again, we wouldn't have been brought into this project if it wasn't for Jason Fish, whose lab we've been collaborating with since we both started our independent groups. And I need to thank Mark Hahn at UPenn for doing the CNS endothelial cremice to us, and this work was paid for by the Department of Defense, as well as American Heart Association, and startup funds from the Cardiovascular Research Institute at Baylor College of Medicine. With that, I'll be happy to take any questions.
Great, thank you so much, Dr. Wythe, for sharing that data with us, and to all of your collaborators for fueling those discoveries. So it looks like we have a question from Aaron. So how long after injection of tamoxifen before you, sorry,
yeah, no
before you took the light sheet microscopy images?
Yeah, so we did a couple of different time points in the paper. The ones I didn't show you is the mice actually die if you do systemic endothelial activation postnatally. Within 14 days, they start dying all the way up to P20. By P21, we have over 50% lethality. So we did it then, and then we also did the CNS one 2 months after they were born. And then we've also taken adult animals and induced it, and then been able to see AVMs 2 months later also in adults, which is very unique for AVM models. So that's the earliest we've looked. We do think it takes time to remodel. So basically we think that hemodynamics actually have a significant role to play. So when we look at the early mice at P14, their vessels are actually kind of hemorrhagic and very thin, and they die. Whereas if we restrict it to just the brain endothelium, then we think that there's enough time and flow and feedback that is closer to what you see in a human. Because the average age of diagnosis for an AVM, BAVM in a human patient is about 22 to 25. So I think there's definitely some hemodynamic feedback that has to happen to really lead to that tortuosity and that large remodeling that has to happen.
So it's the flowing of the blood then that's causing the changes in the modeling and the shape?
We think so, yeah. So we've got some interesting data now looking at autonomous and non-autonomous changes, and it seems like KRAS is actually acting on non-mutant cells to induce effects too, which people have seen in cancer as well. And we definitely think that the flow is really leading to the extensive remodeling and a critical component of it. Yeah.
Thank you.
Yeah.
Awesome. So next question, is the idea that the new KRAS mutant targeting drugs can be used to treat BAVMs?
So that's a great question. So what we're doing right now is from the New England Journal paper and their subsequent studies, the mutations that were analyzed were G12D, G12V, and Q61H. So the drug that Amgen's developed is G12C. So we did not detect that mutation in our patients, but now we've got those mice and constructs in the lab. We're actually testing them to see if they'll develop a AVM. And what's interesting, that mutation is more frequently in like pancreatic cancer than the other two. So we're not sure if it's going to produce the same type of defect in the brain. But if we do see AVMs in that G12C gain of function, then we're actually going to try the Amgen inhibitor and see if we can block it or regress it in the mice. And I'm sure that they're going to be coming out with their next series of drugs that target G12V and D any day now, I would hope, for a variety of reasons. And we would also like to try those. We're also currently trying some KRAS degrader constructs that Terry Rabbitt's lab has generated, where basically they've hooked up specific molecules that recognize either pan-RAS or GTP-RAS or GTP-KRAS to different ubiquitin ligases. So we're trying that using AAV to actually target the endothelium in the hopes that we can basically try and knock down KRAS activity. So we'll see what happens.
Great. So the next question is, any speculation on the mechanism that confers specificity for RAS G12D to activate MEK ERK over PIEK 3K, sorry, 3K AKT?
It's a great question, and we honestly don't know. And now we're getting to the point where we've actually got some MEK mice in the lab as well. And I don't think it's the best allele that we have to try and overactivate MEK signaling, but they don't seem to have AVMs. And we're also trying it with RAF right now too. So my suspicion is that MEK is necessary, but maybe not sufficient. So there's something else. And maybe our assays weren't sensitive enough to find out the role from PI3K in this process, but I'm sure it's much more complicated than we're making it out to be. So, yeah.
Great, thank you. And then the next question is, somatic KRAS G12D drives AVM and cancer progression. Why is KRAS a hot spot for somatic mutations?
It's a great question, and I literally have no idea. We just know from our variant allele frequency, it's got to be happening very late. Because if it was happening earlier, we'd expect a much higher contribution to the endothelium. And we're talking like what we detect by ddPCR is down to like 0.5%. So it's definitely something late. And we also think that the cells seem to actually get walled off and stop proliferating. So there's something weird that happens with them. I didn't really hit on it too much, but the largest category here's angiogenesis, and then like RNA processing and biogenesis and cell size is much, much higher than anything else. We don't have a good handle on what's happening there. And two of the other mutations that we actually identified, so we found 4 hits in KRAS, 1 in P53 and 1 in P10. So I'm sure that there's got to be some kind of similarity to cancer, but they're not over proliferating, which is the really interesting part. Is we think of MEK signaling, you think of overactive proliferation. But in the endothelium, we've done it again and again, and we've done it in the zebrafish, as well as culture cells. It doesn't seem to lead to excessive proliferation. But why? What's so special about the endothelium, and why KRAS is such an activated gene in BAVMs? I don't know.
Great. Thank you, Dr. Wythe.